great to see you all here this evening. So my job is fairly simple. We're not going to break any new ground in panel presentations here this evening. Our three speakers will each talk for several minutes about their work. And when we were organizing this, we asked them all to please give you all their CRISPR origin story, how they got involved in this field. And then I'll ask a few questions, probably myself, but we want to leave a lot of time for questions and answers. And there will be a pop quiz on the video, so I hope you've all paid lots of attention. I know you have their bios in the programs, but in case you don't want to take out your reading glasses, let me give you a short version. And we're going to start with Rachel. Rachel Haurwitz is a co-founder of Caribou Biosciences, where she's been the president and CEO since its inception in 2011. Rachel is also a co-founder of Intellia Therapeutics. In 2014, she was named by Forbes magazine to the 30 under 30 list in science and healthcare. And in 2016, Fortune named her to its 40 under 40 list. Your own personal origin story as it relates to CRISPR, and then of course what Caribou is up to.
Absolutely. Well, good evening, everyone. It's such a pleasure for me to be here with you this evening. It may be hard to believe, but I am a CRISPR old-timer. I have been working on CRISPR since 2007. And as the video outlined, the earliest work in the CRISPR field really came from a very esoteric study of how bacteria and archaea fight off viruses and other selfish pieces of DNA. And so I was involved in that field for several years, and I worked at the University of California, Berkeley, in the laboratory of a woman named Jennifer Doudna. I was the first student in her group to be studying these proteins, trying to understand how they functioned and how they ultimately worked in concert to fight off viruses and allow many different kinds of bacteria to survive. Some of my colleagues in her lab happened to be working on the Cas9 protein that you heard about in the video this evening, which of course holds tremendous potential for genome editing and genome modification. And so I was lucky enough to be part of a group of scientists at UC Berkeley who wanted to found a company, create a space to try to translate a lot of this research into the commercial world. Also lucky enough that all of my co-founders are academics through and didn't want to leave the laboratory. And they gave me the opportunity to try to build and grow the team. And so at Caribou, we've been working on CRISPR gene editing for almost 7 years now. And today, we're primarily focused on developing new medicines using this technology. And we work in 2 different kinds of cells to do that. In part of our work, we modify immune cells, such as T cells or other part of the immune system, using gene editing to tweak these cells to actually teach them how to find and fight cancer. There are versions of these kinds of therapies already approved. In fact, the first ever cell therapies approved in the United States are an earlier version of this type of medicine, and they're called CAR-T therapies. But the versions that exist today, while miraculous for some of the patients who benefited from them, are very challenging to make. It actually requires clinicians to take a cancer patient's own immune cells, their own T cells, out of the body, into the laboratory, manufacture one product for that one patient, and then ultimately re-deliver it back to that patient. It's very challenging. These are very ill people. These cells are not in great shape to begin with. And unfortunately, some of the patients are not even able to live long enough for the manufacturing process to be completed before they're able to receive their therapy. And so at Caribou, along with many other groups, we're working on ways to use gene editing to create the next version of these cellular therapies that can be so-called off-the-shelf, meaning they don't have to be made from a patient's own cells, but with the use of gene editing, we can delete the parts of the cell that would otherwise cause graft versus host disease or some of the other stumbling blocks that currently prevent the field from making these medicines more broadly available. The other part of our therapeutic focus is what we call bugs as drugs. and that is by focusing on the microbiome, which is the collection of microorganisms that live in and on us. And we're particularly interested in some of the bacteria that live in us and in our guts, and the role that they play in how our immune systems fight cancer. And so you can tell that there's a theme in our work of really focusing specifically on cancer therapies. And we're also using gene editing to manipulate microbes that live inside of us in order to better fight cancer. I will highlight, we do get to work outside of the human healthcare space as well. Gene editing has the potential to impact a number of very broad different industries, such as food, agriculture, basic research, and industrial biotechnology. And through licensing and business development, our organization has partnered with a number of companies who use CRISPR gene editing in other spaces, such as plant breeding and even livestock breeding. So one of the projects that we're working on right now with a British company called Genus PLC is actually breeding pigs that are resistant to a particular virus. There's one virus, PRRS is the acronym for the name of the virus, that's the worst problem that American and European pig farmers deal with. And it's an unfortunate situation where if one animal gets sick, basically every animal on the farm will get sick. They're too sickly to then go into the food stream. They all have to be slaughtered, and part of what makes them sick prevents them from reproducing. And so all of the animals are killed, they're not going into the food stream, and a farmer has to completely rebuild his or her herd. And it turns out how this virus infects pigs is well known and has been known for decades. And it's one protein that sits on the outside of certain cells that the virus attaches to, and that's how it sneaks in and infects the pigs. And so what we're doing with gene editing is just deleting that one gene that makes that one protein from the pig genome so that the virus has no way to sneak into these cells. And the work that we and our collaborators primarily have done so far suggests that this is probably going to work. So it's certainly a very new area in terms of science, but we're very excited about it.
Thank you. So Rachel, on my more cynical days, I think that the press is enamored of CRISPR because of the name. But then when I think about it, I realize that in fact it allows scientists to alter genomes in a way that previous technologies did, but much more slowly, much more kludgely, et cetera. What is it about CRISPR that has made it take off in a way that, for the scientists in the audience, zinc fingers and talons just kind of didn't?
Yeah, that's a great point, Sharon. CRISPR is not the first gene editing technology. Gene editing has been around for probably a couple of decades, depending on how you count. But up until CRISPR, you really needed to have a PhD in genome editing to do genome editing. The ways that the scientific community could manipulate the DNA were just a lot more complicated. You had to design a bespoke protein for each new site that you wanted to go after. And there were, and still are, only a very small number of people in the world who have that expertise. And so it meant you either had to be one of those people or pay a lot of money to one of those people to be able to do genome editing. So the technology was around, but was not broadly accessible. And I think it created this pent-up energy and lots of ideas for what you could do if you had access to that. And CRISPR, I think, changed everything. This is an overused phrase, but I'll use it anyways. It really democratized genome editing. Because as you saw in the video, it's quite simple to reprogram the system. The big lump of protein called Cas9 is the same in every experiment. So you can just buy a lot of that, put it in the freezer. You're not constantly making a new protein. And to target a new site in the genome, meaning to edit a new site in the genome, you just have to design a new piece of RNA, which if you're an academic researcher in a lab, you can order from a company custom. It'll show up in a couple of days, and depending on how you ordered it, it costs you tens to hundreds of dollars. And so all of a sudden, this became accessible to a much broader research community.
So we're going to circle back to the question of whether democratizing genome editing is an unadulterated good. But before we do that, let me ask Rachel one more thing about the pig work. So by CRISPRing out the porcine viruses, the PERVs, as you pointed out, that could save herds and could be a huge benefit to livestock producers. A couple of decades ago, when there was some hope for even discussion about raising pigs as organ donors for humans. The FDA shut that down because of the concern about the porcine viruses that could, in the liver, in the lungs, in the heart, whatever, if transferred to a human recipient, could cause very deleterious consequences. So you described the pig work in a livestock setting, but I'm wondering if the technology, the approach that you're describing, could also turn pigs into a source of donors for people who are otherwise on the waiting list forever.
It's a very good question. I'll say it's not something that we or our partners are working on, but I'm certainly aware that there are others who are using CRISPR or other types of gene editing to try to modify pigs for xenotransplantation for exactly what you're describing. I think it's early days, but there are a number of companies who are investing heavily in trying to do this right now.